Back in the day, we used to think proteins transferred genetic material. We knew about nucleic acid, but we didn't know that it was in fact DNA that transferred genetic material. How did we find out? Well, Fred Griffiths in 1928 conducted an experiment that showed that genetic information can be transferred. He found that the 3S strain of Streptococcus pneumoniae, when injected into mice, causes the mice to die, and 3S was recovered, as the immune system could not destroy the S strain. Heat-treated 3S destroyed its capacity to be virulent, which made the mice survive, and no bacteria was recovered, as the immune system was able to destroy it. The 2R strain of Streptococcus pneumoniae, when injected into mice, meant the mice survived. No bacteria were recovered, as the immune system could destroy the R strain. When both the 2R strain and the heat-treated 3S were injected, you would expect the mice to survive, but the mice died, and live 3S was recovered. Griffiths concluded that some heat-stable part of the 3S cells was being transferred to the 2R cells to make them 3S cells. Whatever was transferred must also be inherited, as the recovered cells stayed S when allowed to replicate on a plate. What he found was DNA, but he didn't know it yet. Griffiths termed the transmitted substance the transforming principle. In order to find the transforming principle, Avery, MacLeod, and McCarthy conducted an experiment. They treated heat-killed 3S with techniques to remove RNA, protein, or DNA, so that when one of the variables were removed and replication stopped, they would know that that was the cause of genetic transfer. There was also a control. When DNase was added to the heat-killed S cell, then added to R cells and injected into the living rats, the rats survived and only R cells were recovered. By removing only DNA, did the transformation stop. The scientific community didn't believe Avery, saying that low levels of contaminating protein could have survived the enzyme and that was causing the transformation. To settle the argument between protein and DNA, Hershey and Chase in 1953 used radioactive sulfur 35S to track proteins and radioactive phosphorus 32P to track DNA from a T2 bacteriophage virus to a host cell. The Hershey and Chase experiment involved infecting E. coli cells with the virus, then blending and centrifuging to find out where the protein and phosphorus ended up. The empty phage in the supernatant was where the protein was found, and the DNA was found to enter the cells. Thus, this must be the genetic material. Furthermore, in a second experiment, the phage was given time to reproduce in the bacteria, and the offspring harvested and examined for radioactivity. Small amounts of 32P are found in the new phage, but no 35S. The 32P present in the progeny indicates DNA was passed on, not protein.